So we are going to learn about identification of bacteria. After isolation of a bacteria in pure culture from a specimen, it has to be identified. There are various methods for identification of bacteria, such as first one, conventional method or old method. Second one, automated culture method. Third one, molecular method. We can go in detail to conventional method. Conventional method consists of the specimen subjected to direct microscopy, which is followed by conventional culture of the specimen on blood agar or the selective media, and the colonies which is grown in this agar is taken for culture smear, and the culture smear is followed by biochemical test, motility test, etc. So first one, we can learn the characteristics of a bacterial colony through its morphology. That is, morphology of bacterial colony, that is the appearance of the bacterial colony, and the features studied are size one. First one, size. That is, a pinhead size indicates staphylococci colonies and pinpoint size indicates streptococci colonies. Second one is the shape, and it can be circular or irregular. Third one is the surface. It could be smooth or rough or granular. Fourth one is the elevation. Elevation can be flat, raised, convex, low convex or unpinnate. We could remember this by a term flour. Okay, if you want, it's not necessary. Flour. And fifth one is the edge. It could be entire, crenated, lobate, ciliate or undulate. Sixth one is the opacity. That is, it could be transparent, translucent, opaque. Then seventh one is the hemolysis. That is, hemolysin is an enzyme which causes hemolysis around the colonies. Some bacteria are able to produce this hemolysin. Thus, lysis of RBCs can be seen around the colonies. Eighth one, color. It could be pigment due to the pigment or due to the color of the media. Pigment can be diffusible or non-diffusible. Diffusible means it could be throughout, cannot be differentiated, but non-diffusible means the color is only seen in the colonies, and culture media does not have the color. Ninth one is the consistency. It could be mucoid, friable, firm or buttrons. Then we can go to growth in liquid media. The media used is the peptone water and nutrient growth, and growth can be indicated by 3, that is uniform. If uniform turbidity is seen, then there is gram negative bacteria. If it is a deposit at the bottom, then we could say it's streptococci. Third one, if there is surface pellicle formation, then we can say it could be Pseudomonas. The pellicle formation is due to the increased presence of oxygen in the surface. So we can conclude by telling B, growth in liquid media. The peptone water and nutrient growth provides that's a medium, and if there is uniform turbidity, then we can say it's gram negative bacteria. Second one, if there is deposit at the bottom, then we could say it is streptococci. Third one, if surface pellicle is formed, Pseudomonas. Okay. Next one, staining. Staining mainly 3 types, that is gram staining. So could be, we can differentiate bacteria as gram positive or gram negative bacteria. It is applicable to most of the bacteria. Second one, Albert staining. It is used to stain specific Corynebacterium diphtheriae, And third one is the Sale-Nielsen staining. It is used to differentiate between acid-fast bacilli and non-acid fast bacilli. That is Mycobacterium tuberculosis and Mycobacterium leprae are both acid-fast bacilli. They can be identified by this Sale-Nielsen staining. So staining, Gram staining, Albert staining, Sale-Nielsen staining. Then D, motility testing. It could be with the help of the hanging drop preparation method. Motility testing, that is bacterium is can identify whether bacterium is motile or non-motile. The fifth one is the E, that is the biochemical test. There are various biochemical tests, and in that catalase and oxidase test can be done in all bacteria. Then to identify gram negative bacteria, there is a code, that is I cut. It means I for indole test, C for citrate utilization test, U for urea hydrolysis test, T for triple sugar ion test. Okay, I cut. Indole test, citrate utilization test, urea hydrolysis test, triple sugar ion test. Third one, other tests are also there for further identification of the bacteria. They are sugar fermentation test, methyl red test, Voges Proskauer test, oxidation fermentation test, nitrate reduction test, decarboxylase test, phenyl pyruvic acid test, etc. So we can go in detail to the catalase test. The principle is that certain bacteria has catalase enzyme and which acts on this catalase enzyme acts on hydrogen peroxide to release oxygen. Okay, procedure is pick up the test bacteria with platinum blue from nutrient agar slope or plate. Then mix it in a drop of H2O2, that is 10 volume, on a clean glass slate. Okay, then we can see some observation. Then the test is said to be positive if there is immediate bubbling. If there is immediate bubbling, then we can say that catalase test is positive. And if there is no bubbling, then we can say that catalase test is negative. This bubbling is due to oxygen formation. Then we can say the positive test shown by all members of catalase positive are all members of enterobacteria except Shigella dysentery type 1, then Micrococcus, then Bacillus staphylococcus. So catalase positive are all members of enterobacteria, then Micrococcus, Bacillus and Staphylococcus. Then catalase negative are Shigella dysentery type 1, that is a member of enterobacteria, exception is there. Okay, catalase negative is Shigella dysentery type 1, then Streptococcus, then Clostridium. The main use of this catalase test is to primarily is to differentiate between genera Staphylococcus and Streptococcus. Staphylococcus is catalase positive, while Streptococcus is catalase negative. Then we can go to oxidase test. Oxidase test principle is to determine the presence of enzyme cytochrome oxidase, which can catalyze the oxidation of reduced cytochrome to molecular oxygen. So reduced cytochrome is oxidized to molecular oxygen in the presence of the enzyme cytochrome oxidase. So some bacteria are able to produce the cytochrome oxidase. Then we can identify which type of bacteria. Okay. So the procedure is there is an oxidase reagent, that is freshly prepared solution of 1% tetramethyl paraphenylene diamine dihydrochloride. So the oxidase reagent is freshly prepared solution of 1% tetramethyl paraphenylene diamine dihydrochloride. So the procedure is the filter paper strip soaked in oxidase reagent is smeared with the test organism or bacterium. Okay, or in this, if there is a deep purple within 10 seconds, that is oxidate positive test. Okay, if there is no color change, then the oxidase test is negative. There is another procedure also, that is pouring the oxidase reagent on the surface of the colonies, and if the colonies become purple within 10 to 30 minutes, then also the oxidase test is positive. This method is mainly used to differentiate, that is used to pick up Neisseria colonies from mixed growth on culture media, and it can be subcultured to get the pure culture. Okay, so the use of and the oxidase positive bacteria are Pseudomonas species, Vibrio, and Neisseria species. Okay, oxidase positive are Pseudomonas species, Vibrio, Neisseria species. Then oxidase negative, they are all members of Enterobacteria, Bacteriaceae, and Stenotrophomonas maltophyllum. So oxidase positive is Pseudomonas and oxidase negative is Stenotrophomonas. Okay, Stenotrophomonas maltophyllum. Okay, then we can go to next. Third one is the indole test. Indole test principally tryptophan can be converted into indole with the help of the enzyme tryptophanase. So the reagent is Kovacs reagent. So the interpretation is, if the test is positive, if the indole test is positive, then there is red colored ring near the surface of the medium. Then this positive test is shown by Escherichia coli, Proteus species, and Edward Silla. Okay, and the negative test shows yellow colored ring near the surface of the medium. Yellow colored ring near the surface of the medium, and it is shown by Klebsiella species and Proteus mirabilis. So we said positive indole test is shown by Proteus species except Proteus mirabilis. So negative test of indole test is shown by Proteus mirabilis and Klebsiella species. Fourth test is citrate utilization test. Principal ability of an organism to utilize the citrate and salt source of carbon as a sole source of carbon for its growth with resulting alkalinity. That is, the reagent the medium used are Simon citrate medium, that is solid medium, and Koso citrate medium, that is the liquid medium. So interpretations are, in Simon citrate medium, positive citrate utilization test is shown by the intense blue color on the slant. It is shown and the negative Simon citrate test is, there is no growth, so there is no change in color, that is the medium stays green. And the Koser citrate medium, the positive test is seen by turbidity due to the growth, and negative is negative is understood by no turbidity. So the citrate positive organisms are Klebsiella and Salmonella species, and citrate negative are Salmonella typhi and Escherichia coli. Okay, next we can go to urease production or urea hydrolysis. Principle is ability of an organism to produce the urease enzyme. This urea can be converted into ammonia with the help of the enzyme urease. The ammonia is alkaline, which gives an alkaline medium. So if we are adding phenyl red in the alkaline medium, the phenyl red changes its color to phenyl pink or red. So the medium used is Christensen's urease medium, and the interpretation is if it is positive, that is the urease production or urea hydrolysis test is positive, then there is pink color formation, and it is shown by the species such as Klebsiella species, Proteus species, Helicobacter pylori, Yersinia enterocolitica. And negative test is shown by the pale yellow color formation. Example is that is shown by the species like Escherichia coli, Providencia species, Yersinia pestis. So Yersinia enterocolytica is showing positive urea hydrolysis, while Yersinia pestis is showing negative urea hydrolysis. Next test is triple sugar ion test. From this test we can get three conclusions. That is the ability of an organism to attack a specific carbohydrate in the growth medium and to form acid. Second one is that formation of acid can be with or without production of gas, and third one is H2S is also, that is hydrogen sulfide, is also produced along. So the TSI medium consists of sugars like glucose, lactose, sucrose and ferric salts, so that H2S can be identified, and phenol red for indication of the acid formation. So interpretation are, if there is yellow color, then there is the medium is acidic, that is fermentation has occurred. So fermentation of carbohydrate has occurred. Then if the medium is red in color, it is alkaline, so there is no fermentation. Then if there is bubbles in the butt, then we can conclude that gas is produced. And if there is blackening of the medium, then there is H2S production. This blackening of the medium is produced by Proteus mirabilis, Proteus vulgaris and Salmonella species. With there is exception, that is Salmonella paratyphi. Okay, that is H2S is not produced during fermentation of carbohydrate by the Salmonella paratyphi and Marginella species. Okay, that is H2S production is negative. It's not shown by Salmonella paratyphi and Morganella species. Okay. Next we can go to sugar fermentation. Sugar fermentation principle is the ability of an organism to ferment specific carbohydrate in the medium, producing acid or acid with gas. So interpretation is that if the test is positive, then there is pinkish red color, that is the acidic nature is shown. The indicator is Andreid's indicator, and this pinkish red color is given by fermenters. That is there are different types of fermenters. First one is glucose fermenters. That is example of glucose fermenters are all members of Enterobacteriaceae. Then glucose and lactose fermenters, that is Escherichia coli and Klebsiella species. Glucose and mannitol fermenters are Salmonella species. If the test is negative, then there is yellow, then the yellow color changes to colorless, that is colorless shows alkaline medium. If gas production is there, it can be seen with the help of Durham's tube. The Durham's tube will be inverted, so the bubble formation can be seen. So next test is methyl red test. The principle is production of sufficient acid during the fermentation of glucose, and the second one is the sustained maintenance of pH less than 4.5. So interpretation is that if it is positive, methyl red test is positive, then there is a red color formation and red color, and it is given by this test is positive by Escherichia coli, Yersinia species and Listeria monocytogenes. Okay. Then the test is negative by the formation of yellow color, and it is given by test is negative by species such as Klebsiella species and Enterobacter species. Klebsiella and Enterobacter species. Okay. Then next test is Vogt-Proskauer test, VP. Okay, VP test. Principle is pyruvic acid is converted into acetyl methyl carbinol or acetoin. This acetoin undergo oxidation in the presence of atmospheric oxygen and alkali to give diacetyl. This diacetyl combines with alpha naphthol to give a red color. So if the test is positive, there is pink color in 2 to 5 minutes and it deepens to crimson in 30 minutes. This test is positive by Klebsiella species, Enterobacter species and Staphylococcus. And the test is negative, means there is color, it is colorless for 30 minutes. There is no the medium is colorless for 30 minutes. It is negative by Escherichia coli and Micrococcus. This the PP test is not given by Escherichia coli or Micrococcus. Then we have next test, that is the nitrate reduction test. Principle is reduction of nitrate, or that is reduction of nitrate into nitrite or free nitrogen in the presence of nitrate reductase. Interpretation is that positive red color develops within few minutes. All members of Enterobacteriaceae or Barhamella catarrhalis, okay, all members of Enterobacteriaceae, okay. And negative means there is no color development and Haemophilus ducreae, okay. Next test is phenylalanine deaminase or phenyl pyruvic acid test, PPA test, okay. Principle is deamination of phenylalanine into phenyl pyruvic acid. So interpretation, if it is positive, green color and it is given by Proteus species, Morganella species and Providencia species. If it is negative, there is no color change and it is given by all members of Enterobacteriaceae. So PPA test is negative by all members of Enterobacteriaceae. Then there is the oxidation fermentation test. In this principle, acid production changes color of medium from green to yellow. So if the sugar is fermentatively utilized, there is acid produced in the both tubes. So there are two tubes and acid will be produced in both of them. In one of the tube there is paraffin coating, so there is anaerobic condition. If there is non-fermentation that utilize sugar oxidatively, color change only occurs in the tube without the paraffin overlay. And acrolytic bacteria cannot utilize sugar in both tubes, so there is no color change in both. So next we can go to, that is the antigenic structure. By using specific sera, we can identify organisms by agglutination reaction, slide agglutination test, okay. That is, example is Salmonella is confirmed by agglutination with specific antisera. Next one is the typing method. Typing method can be of two types, that is phenotypic typing and genotypic typing. Examples of phenotypic typing, they are used for intraspecific differentiation, and example are bacteriophage typing, bacteriocin typing, etc. Genotypic typing includes plasmid profile analysis, ribotyping, etc. Now we can go to pathogenicity. In pathogenicity means inoculation of test organism into lab animals like guinea pig, rabbit, rat, mouse, etc. by intradermal, intramuscular, intravenous, intracerebral, intraperitoneal or intravenous or oral or nasal spray, okay. Then antibiotic, next we can go to antibiotic sensitivity test, okay. The isolated bacterium subjected to antibiotic sensitivity test in vitro for selecting appropriate antibiotic for therapeutic use, okay. Then we can go to the second method for identification of bacteria is the automated system for bacterial identification. The examples are MALD-TOF, that is a matrix assisted laser desorption or ionization time of flight method, and VITEK2, VITEK2. The third one is the molecular method It includes it is broadly classified into amplification methods and non-amplification method. The amplification method include PCR, that is polymerase chain reaction, and other one is real-time PCR, that is real-time polymerase chain reaction. Then the non-amplification is the DNA hybridization. It includes the process of DNA hybridization, that is the line probe assay method. Okay, thank you. That's all for today's class.